Good morning, everyone. I'm really happy to be here with you today, presenting my master's work for the first time. My name is Sage, and I'm a student from the University of Guelph. And today I'll be sharing with you my research on cavity nesting bees across Canada using the community science program Bees at Schools. And so I don't think I need to convince anybody here on why it's important to study bees, but bees provide essential pollination services and huge economical growth to our crops, as well as huge value to our natural ecosystems. Unfortunately, our native bee populations are threatened by a lot of things, climate change, habitat loss, and non-native species competition, just to name a couple. I'm sure you know more of them. So in Canada, we have incredible bee biodiversity, over 900 species. For this study, we chose to focus on cavity nesting bees. Cavity nesting bees are solitary bees who use hollow plant stems, holes in wood, or in metal grating, as you can see here, to build their nests. And so the females will each make their own nests in rows like the photo on the bottom. Cavity nesting bees are really important pollinators for many crops and wild ecosystems. And they're also found to be representative for the biodiversity of nest building bees as a whole. So to a point, they can be representative of the conditions for bees on a larger scale. Now, as I mentioned, bees are threatened by a few things right now, and habitat loss and degradation is one of the main ones. Humans have a huge impact on the ecosystems around us, and areas like mass agriculture and urban areas really change the biodiversity. So there have been quite a few studies on bees in urban areas, and so far they've found conflicting results. Some find that urban areas are positive for bee populations, and some find that they're negative. So further research into this is necessary so we can learn how to help them best. So with all this in mind, I have a few questions that I'm looking to answer here. So the first is, what exactly is the distribution of cavity nesting bees across Canada, and how are they affected by the local landscape? I used a landscape of 500 meters. What are these cavity nesting bees using for food to feed their offspring in their nests? And how are cavity nesting bees interacting with their omnivorous cousins, the cavity nesting wasps? So to answer these questions, I used the Bees at Schools community science program. So a community science program uses the general public to collect their data. In this case, we sent these nest boxes here to schools all across Canada, and we had the teachers and their classes install them. We had teachers install them in late April to early May and leave them up until the end of August, which is the flight period for most cavity nesting bees. Now, our 2020 sampling season had a unique challenge in that schools were closed right when we were trying to send out our nest boxes. Luckily, many of these incredible teachers pivoted and installed the nests in their home properties and shared videos with their students, etc. It's really awesome. So trap nests or bee hotels or nest boxes have been used as a sampling method for decades. Our particular trap nests were using Dr. Scott McIver's design, which has been used in some studies to good success so far. So each nest had 30 cardboard tubes, 10 each of 3 different sizes. Now, because we outsourced the installation of these nests, we gave parameters to the teachers so that they could more easily find a good location to install them. So our nests were installed between 0.7 and 2.5 meters off the ground, and most of them were installed facing south. So here we can see a map of all of our successful installation and returns. The one challenge with using teachers and schools as our samplers is that we can only send nests where there are teachers willing to receive them. So you can see we have a fair number of nests in Southern Ontario, Southern BC, etc. Once the nests were shipped back to us, it was time to get to work in the lab. So many studies which use trap nests then hatch out the occupants in order to identify them. But in our case, we also wanted to look at what pollen the bees use to provision their offspring. And so pollen is notoriously difficult to identify taxonomically, and the longer the larvae are alive, the more they consume. So instead of hatching out our occupants, we used DNA metabarcoding to identify them and the pollen they were eating. So our first step here was to remove the larvae from the tube. I did this by cutting them open, and that's about it. Each tube itself was our sample unit instead of using each small leaf packet or soil packet, because that would have just been far too many samples to deal with. Once all of the nests were open, then came the DNA barcoding portion. And so DNA barcoding uses a short segment of DNA that is species specific to compare to a reference library of known species. And so we use DNA metabarcoding, which uses the same method, but can be done on multiple species in the same sample. So we were able to find examples where there were multiple types of pollen used by one bee, or multiple bees using the same tube. So once all the DNA extractions were done, we were able to sequence them. We used the CO1 marker for the insects, bees, and wasps, and the RBCL marker for our plant identification. So once all of this was finished, I ended up with some massive spreadsheets, and I'm still working on going through my results, but I have some preliminary stuff that I'd like to share with you. So our first question was looking at the distribution of these bees across Canada. Now this is the same map, our site map, but here the blue dots represent nests that had some occupation in them. And the size of the dot represents how many tubes in that nest were occupied. So I do find it interesting, even though we had so many nests, say in Southern Ontario, and some of them fairly close together, there was still obviously something driving those that were occupied versus those that were unoccupied. So in 2020, we had 4 times the number of successful nest installations. Now we sent out double the nests in 2020. So part of the success is due to sharing better instructions, as well as being on email quite a bit. So despite having 4 times the number of successful installations, the nest occupancy rate and tube occupancy rate stayed very stable. So I found this pretty interesting, and I'm curious to see if this year's 2021 data will reflect that same pattern. And so these are all of the bee species that we found in our nest boxes in 2019 and 2020. So there's a few things that I'd like to point out. First is Megachile rotundata, our #1 most found species. Now this is an introduced species used for agriculture. Found that one quite a bit. Our second most common species is Osmia lignaria. This is a native species to Canada. And our third most found species is Osmia taurus, which is also a non-native species. Now we also found some kleptoparasitic bees, which are pretty interesting. They don't make their own nests. Instead, they'll lay their eggs on a previously made nest, and their eggs will use up all the resources. So I thought it was interesting to look at who they were hosted by. So Coelioxys funeraria was hosted by 3 different bees. It was found in the same nest as 3 bees. Coelioxys moesta was found in the same box as 3 other bees. Coelioxys speculifera was found in the same box as 2 other bees. And our 4th kleptoparasite, Stelis vernalis, was found only with 1 other host. So I found that pretty interesting. These hosts seem to line up with previous hosts in the literature. And the last thing that I'd like to point out is that we had some pretty obvious community turnover here. In 2019, there was a large number of other bees. You know, we've got some Hylaeus, Hyraeades here, which we did not find in 2020. Whereas in 2020, we had a very large number of our Osmia, our mason bees. So we don't quite know why that is yet. We're going to keep looking into that. And now here we have a PCA of our landscapes for 2019 and 2020 sites. The landscapes were found using the 2015 Landcurve Canada data, which is a 30-meter resolution. And so we were pretty happy to see that we had a good spread of agricultural versus forested versus urban sites. And so we were able to use that in ongoing analysis. And we have just begun looking into these, but so far, here we can see our bee species richnesses in each of our sites, beginning with crop, forest in the middle, and urban at the end. And so in 2019, although not significant, it does appear that cropped habitats had a slightly higher richness. However, in 2020, all of the sites and their landscape types appeared to have fairly even richnesses. So we're going to continue looking into this as well. Now, our second question was looking into what these cavity nesting bees are using for food. So we're still waiting for our 2020 data to come back for this. So this is just our 2019 preliminary work. So we can see that a lot of bees were sampling from a lot of plant families. I was kind of expecting to see some kind of specialist behavior here, but it's not evident in this graph. To look into it further, we reduced it down to the top 5 plant families to see if that would reveal any specialist behavior, but it didn't really seem to. These bees all seem to be acting as generalists, which would make sense because they're living in areas in close proximity to humans. And so, you know, as Kit was saying, these areas have a lot of non-native plant species and species that the bees wouldn't be involved with. So generalist species tend to do better. Now, our third question is going to look into how cavity nesting bees and wasps are interacting. And I am still just looking into this one. This is a very challenging question, I'm finding, because cavity nesting wasps are less investigated than bees. And we have wasps, some of which are kleptoparasites, using the nests of other bees and wasps. And we have some that are just predators and make their own nests similarly to bees. So I'm having difficulty figuring out which kind of wasp is which. If anybody has any good wasp resources, let me know. And so I have a lot of next steps. I've just began looking into my data. And so, of course, I'll be looking at this 2020 plant data when it comes back. I expect to have a much larger bipartite network to look at and hopefully maybe see some specialists. We'll see. And then, of course, I'll be looking into how the cavity nesting bees and wasps are interacting. The other cool thing is that the Bees at Schools program ran for its 3rd year this year, and that data is currently being looked at by another master's student. So the more data we collect, hopefully the more patterns we'll be able to see. So what about this is important? Well, community science is a super powerful data collection tool. This data could not be collected without all the teachers and students. And so despite all the challenges with this method, it was great. Now, we saw that the bee community structure changed between 2019 and 2020. Even though we had so many more samples in 2020, the number of species that we detected did not go up. So I think that's really interesting, and we need to look into that further. And also, these bees living close to humans feed from really diverse pollen sources. They're not specialists, at least not the ones we've seen yet. So I want to send a shout out to the many, many folks without whom this would not have been possible. The Steinke and Raine labs for teaching me all about things, and my friends and family who spent hours building and shipping nest boxes. So thanks so much for listening. My email and Twitter are on the bottom of the slide in case you have questions later on, and I'm happy to take questions or answer them now if we have time.
Great. Thank you so much, Sage. That was great. And we look forward to seeing what you do in the future. So we will open it up, for questions. I think we have time for at least one here. If anybody wants to put a hand up, we can hear a new voice, or I will go into the question answer box. So it does seem Amro has a question. He has his hand up if he'd like to unmute.
Oh, yes. Thank you. Great talk. I was wondering why you picked RBC and not like ITS for the plant barcode.
Great question. We actually ran both RBCL and ITS for our 2019 data and compared them after that. And the ITS had so many markers from so many other things, all the fungus that was also found in the nests, that the signal didn't work out as well. So we stuck just to RBCL for the 2020 data.
Okay, excellent. Thank you.
Thanks.
Great. So again, I wish we had time to get to all the lovely questions, but if you want to, Sage, pop into the question and answer box if you have time and maybe type out some answers for everybody, that'd be great. And thank you again.
Thanks.